Welcome to G Fun Facts Online, the show that dives deep into the wonders of science and the world around us. Today, we're embarking on an incredible journey into the microscopic, into the very heart of our cells. We're going to unravel the mysteries of something called cellular condensates. It's a dynamic dance happening within you right now, and understanding it is revolutionizing how we see health and disease. Get ready, because this is a game changer in biology. For a long time, we thought of cells as tiny bags with well-defined compartments, like the nucleus or mitochondria, all neatly wrapped in membranes. But imagine finding out there's a whole other layer of organization, bustling activity hubs that appear and disappear as needed, all without any membranes at all. These are cellular condensates, also known as membraneless organelles. Think of them as the cell's pop-up shops or flash mobs, concentrating specific molecules to get important jobs done quickly and efficiently. So, how do these living droplets form? It's a fascinating physical process called liquid-liquid phase separation. Picture this: you mix oil and vinegar. They don't blend, right? They separate into distinct droplets. Something similar happens inside our cells. Specific proteins, especially those with flexible sticky parts called intrinsically disordered regions, or IDRs, along with RNA, can spontaneously team up. These IDRs act like molecular Velcro. When enough of these weak, sticky interactions happen, they pull together, forming a dense, droplet-like structure, a condensate, separate from the rest of the cellular fluid. It's a bit like a molecular party where all the cool molecules gather in one spot. And these aren't static blobs. They are incredibly dynamic, constantly exchanging members with their surroundings, assembling and disassembling in response to the cell's needs. The formation and behavior of these condensates are governed by some cool biophysical principles. We've mentioned the stickiness from IDRs and the crucial role of multivalency, needing multiple interaction points. RNA isn't just a passenger, it's an active player, acting as a scaffold, influencing condensate size, and even their breakdown. The material properties of these condensates can also vary, from being very fluid like water to more gel-like, and this state is vital for their function. Of course, the concentration of the molecules involved and environmental factors like stress or temperature also play a huge part. Now, why should we care? Because these condensates are the unsung heroes orchestrating much of cellular life. They're involved in gene expression, helping to turn genes on or off. They manage RNA, deciding which messages get translated into proteins and which get stored or degraded. Think of P-bodies and stress granules, which help cells cope with tough times. The nucleolus, probably the most famous membraneless organelle, is where our ribosomes, the cell's protein factories, are made. Condensates also streamline signal transduction, making cellular communication more efficient, and they even help repair damaged DNA by concentrating the repair machinery right where it's needed. They can buffer cellular noise, and, incredibly, recent research suggests their formation can have global effects on the cell, far beyond their immediate location. But what happens when this perfectly choreographed dance goes wrong? When order turns to disorder, the dysregulation of cellular condensates is increasingly linked to a wide range of human diseases. This is where things get really critical. In neurodegenerative diseases like ALS, frontotemporal dementia, Alzheimer's, and Parkinson's, the connection is stark. Mutations in proteins that form condensates can make them go from dynamic liquid droplets to more persistent, solid-like, and potentially toxic aggregates. Imagine that molecular party turning into a frozen, unhelpful clump. Very recent research even suggests a complex role where the condensates themselves might initially try to protect against harmful protein aggregation, but disease mutations can mess this up. Cancer is another major area. Aberrant condensates can mess with gene expression, driving tumor growth. They can disrupt signaling pathways that control cell division, leading to the uncontrolled proliferation seen in cancer. They're also implicated in DNA damage repair, and defects here can cause genome instability, a hallmark of cancer. Some studies even link them to how cancer spreads and resists treatment, and it's not just neurodegeneration in cancer. Viruses are masters at hijacking our cellular machinery, and that includes manipulating condensates to help themselves replicate. Dysfunctional condensates are also being explored in cardiovascular diseases, aging, and metabolic disorders. The problem can be that condensates form when they should, don't form when they should, change from helpful liquids to harmful solids, or trap essential molecules so they can't do their jobs. This might sound a bit daunting, but the good news is that the field of condensate research is absolutely exploding with new discoveries and hope. Scientists are developing incredible tools, like super-resolution microscopy and optogenetics, using light to control condensate formation in living cells. There's even a new technique called LITEC for studying these structures without disturbing them. Researchers are working to decode the molecular grammar, the rules that dictate how condensates form and function. And most excitingly, this understanding is opening up brand new therapeutic avenues. Imagine drugs that could dissolve harmful condensates in neurodegenerative diseases, or stabilize beneficial ones. Scientists are exploring ways to prevent that liquid-to-solid transition, or to specifically target the interactions that form disease-related condensates. There are even innovative approaches, like RIBOTACs and PROTACs, being adapted to remove problematic RNA or protein components from condensates. Those once undruggable, intrinsically disordered regions are now becoming viable targets for new medicines. So the discovery of cellular condensates has truly opened a new chapter in cell biology. These dynamic, membraneless droplets, formed by liquid-liquid phase separation, are active orchestrators of almost every aspect of cellular life. From keeping us healthy to playing a role in some of our most challenging diseases, their influence is profound. While there's still so much to learn, the rapid pace of research is incredibly promising. Understanding the ins and outs of these living droplets will undoubtedly lead to new ways to diagnose and treat a host of human conditions. It's a compelling and evolving story, reshaping modern biology right before our eyes. Thanks for tuning in to G Fun Facts Online. We hope you're as fascinated by the dynamic dance of cellular condensates as we are. Join us next time for more amazing insights into the world of science. Stay curious.